Welcome back, friends. In this video tutorial we'll be talking about Pseudomonas species. Okay, so let's talk about the properties of Pseudomonas species bacteria. Now, among the whole Pseudomonas species bacteria, we are interested in a particular strain, and that is Pseudomonas aeruginosa, because among the whole lot of Pseudomonas in this total Pseudomonas genus, there is the only one bacteria which is of human clinical significance, because that Pseudomonas aeruginosa is causing dangerous diseases in human being. Otherwise, other Pseudomonas species are good, they're fine to be stayed in your health, in your body, so no problem about that, right? Now, usually Pseudomonas species are gram-negative, so let me write, they are again a part of gram-negative bacteria. So let me write, they're a part of gram-negative bacteria, and they are encapsulated, so they are having a capsule, so they are encapsulated motile, let me write, motile rods. They are encapsulated motile rods. So if I draw the structure, how are they looking like? They look something like this. So let's say this is a bacteria, it's a rod-shaped bacteria, and they are having a capsule outside. So the capsule is there, it is made up with a kind of carbohydrate structure, and they are also having a flagella coming out from a pole. So they are polar flagellate type, right? That means they are having a flagella. So let me write, there is a flagella from a pole, right? So from a pole, so it's a polar flagellate. So as they are having flagella, they are motile, or they are having the movement. So that's why I've written motile and rods, because they are rod-shaped bacteria, so they are rods and motile in nature. And then oxygen requirement, we can tell that they are of obligate aerobic type, so they must require oxygen for their growth. Okay, and the important and unique thing about this bacteria, that this bacteria can produce a kind of diffusible green and blue pigments. That's the important thing. What they produce is a kind of pigment. So let me draw. So let's say green or blue. So let's say blue. So they can produce a kind of this green, blue pigment, which are diffusible. These pigments are diffusible. Diffusible means when we are culturing this bacteria in different agar plates or petri plates, we are going to see that these green and blue materials or green and blue pigments are generated, and they are diffused through from those colonies, right? So let me draw here. Let's say this is the agar plate. So this is the petri plate having the agar, containing the agar or medium for this bacterial growth. Now we can find the growth of this individual bacterial colony like that, and what we can find is that surrounding these colonies there is a kind of slight bluish molecules, or slight bluish chemicals that are coming out, or slight greenish molecules or chemicals coming out in partial region. So these molecules or pigments can diffuse through, which is making a slightly green and blue appearance surrounding the colony, right? So we can find this kind of effect there. So that's the uniqueness about the Pseudomonas species colony. Now we can culture them in laboratory using very simple kind of media, because this is another important point about this Pseudomonas aeruginosa and Pseudomonas species, is that they are very low nutrient requiring. So very low nutrient requiring bacteria, right? So they won't require that much of nutrient sources for their growth. Very few will do the trick and all this thing, right? So that's a very important concept. And also another very important positive side of these microorganisms are that they are kind of heat tolerant. That means in hot tub in laboratory, in water bath in laboratory, usually the temperature reaches 70-80 degrees Celsius, even 100, even slight below than 100 degrees Celsius temperature. This kind of bacteria can still live there for a longer period of time. So if we do not disinfect our materials in perfect time, or through the perfect duration, this kind of bacterial contamination will be there throughout the time. So that's a very challenging task, to remove this Pseudomonas bacteria from our laboratory equipments, and also very importantly that for that surgical instruments, right? Because surgical instruments are reusable, because some surgical instruments are use and throw, but some which are scalpels and all the scissors and everything, which are required in operation, after operation, and all these things. So we do not just throw them away, because we require them throughout the time, so we need to disinfect them. Now for those surgical instruments, if we are unable to disinfect them properly through a longer period of time at a particular temperature, this kind of bacteria like Pseudomonas aeruginosa will be living there, and they can cause infections. So that's why Pseudomonas aeruginosa, so let me write Pseudomonas aeruginosa or other Pseudomonas species, especially Pseudomonas aeruginosa, this can cause mostly nosocomial infections. Nosocomial infections. Now, nosocomial infection means infections occupied or occurred or occurred from the hospital sources, right? So, infections occurred uh, from the contaminated sources in hospital, right? So if you are admitted in the hospital, you are diagnosed with a particular disease, you are getting treatment from the hospital, but during this particular kind or surgical timing, if there is any trace of Pseudomonas species in surgical equipments and all this thing, they can be transferred into your body and they can cause infections, right? So this type of infections are called nosocomial infections, and we are going to see there are a lot of nosocomial infections associated with this Pseudomonas species, because they can live and survive up to a particular heat tolerance level. So they are not usually destroyed using heat and all these things. And another important thing is that they require very low nutrient for their growth. So very small nutrient, if they get, they start to grow. That's a very good advantage for them and disadvantage for us, right? Okay. And about their carbohydrate fermenting ability, what we know is that they can oxidize carbohydrates. So let me write, they can oxidize carbohydrate, but cannot ferment it. So they cannot ferment lactose. So they are actually lactose non-fermenting or lac minus type of bacteria. And if you want to develop the culture in our laboratory, we may use McConkey agar. Remember, we can also use McConkey agar for the growth of E. coli. So we can also use here the McConkey agar. Okay. But again, we need to be very careful for the hospital users, because this can spread from the equipments to our body in hospitals, right? So these are very much related in those cases. Okay, so among the rest of the part, we'll be talking about Pseudomonas aeruginosa, because the Pseudomonas aeruginosa, which can be transferred from different mode of regions in hospital, from hospital or from household apparatus, that can be transferred into our body. Now, as they are transferred into our body, they can cause several rounds of infection, because remember, these infections are from the nosocomial site, or from the hospital lines or from the hospitals. So as a result of that, if the infection, it is related with, say if the surgical procedure is going on in your chest, so it will be related with the infection at your chest region. If the surgical approach is through your lungs, then it will be related to your respiratory tract infections. If it is related with your digestive system or stomach, then it will be related with your digestive system. So there can be many regions of your body that can be infected by Pseudomonas species, especially Pseudomonas aeruginosa, right? And especially the Pseudomonas aeruginosa present in air all the time. It's a kind of organism that air carries with it. So that's why the term comes as aeruginosa. And also they are aerobic, so that's another reason for that naming aeruginosa, and obligate aerobic. So if we put them in very micro oxygen containing environment, we can get rid of this kind of bacteria. So that's a very good side, because as they are obligate aerobes, we can go against them by putting them in micro aerobic micro oxygen environments. Okay, but again, we need to be very careful about this, because they can cause several rounds of infections in GI tract, infection in say uh, in our lungs, which is related with bronchitis, and also they are related with infections in brain like meningitis. Okay, meningitis. So all these things are linked. All this infection can be done by Pseudomonas aeruginosa because they are not having any kind of specific mode of infection, because whenever they get some entry they will enter there and cause diseases, right? So that's it. So we are going to study the infectivity in the future video. Thank you. Okay guys, so we already seen the introductory video about the Pseudomonas aeruginosa. Now in this video we'll be talking about the infection, right? Now one thing about Pseudomonas aeruginosa I must tell you first, that Pseudomonas aeruginosa is not a kind of deadly pathogen all the time. Instead they are opportunistic pathogen, right? Opportunistic. So let me write opportunistic pathogen, right? So what do we mean by opportunistic pathogen? Opportunistic pathogen means this kind of bacteria usually normally present as a microflora in our body at a particular region. In normal conditions they won't infect us, they won't cause any disease, but after a certain time when the environment change to their favor, they start to infect us and causing disease. So those microorganisms are called opportunistic pathogen, right? Now for these microorganisms like Pseudomonas aeruginosa, which is a kind of opportunistic pathogen, they are present almost everywhere in our bodies. Like they are present in our skin, they are present in usually they are present in our skin, because skin is acting as a barrier, right? First important barrier against all the pathogens. Now if there is a cut in skin, if there is a cut, any kind of cut in skin, it will enter into our bloodstream, and whenever it enters into our bloodstream it will be transferred from one place to another place via blood, and then it will cause disease. But again, it cannot cause diseases, it usually not cause diseases, but if there is a kind of compromised situation, if there is the infectivity of other organisms, other microorganisms, or any kind of diseased condition in body, in a particular tissue which is getting weak already, in those conditions they start to take the advantage and causing further more infections. That is the infectivity mode of Pseudomonas species, especially Pseudomonas aeruginosa. Okay, now usually what we can see, that usually this Pseudomonas aeruginosa can be causing different diseases. So let me write the different zones of disease. So the disease can be there. So let's take, the disease can be caused in skin, simple skin disease. Skin disease, among the skin disease what we can get, among the skin disease we can get, let's say we can get rashes, we can get pustules, okay, and skin rashes, pustules, and simple conditions like that. Or sometimes skin is kind of damaging and just coming out, these things will occur. Now this can be related with eye. Now when it is related with eye, we can have infections like, let's say we can have infections like kind of uh, ophthalmic ophthalmitis. Okay, we can have kind of ophthalmitis, and also we can have diseases in conjunctiva. So it can infect conjunctiva, or that means conjunctivitis. So let me write conjunctivitis will result after the infection of Pseudomonas aeruginosa in our conjunctiva, right? So in eye it can cause this kind of disease. Now it can also cause disease in, let's say it can cause disease in our respiratory system, like in lungs or regions like bronchus and all these things which are related with this respiratory system. So it can cause a form of bronchitis. So let me write, it can cause a form of bronchitis, okay, and it can also cause disease, it can cause symptoms, let's say in brain, right? So let me write, it can cause also disease in brain. Now usually the brain disease that it causes is meningitis, and it is common in infants, otherwise we won't see this. So meningitis, okay. So you can see it can infect several regions, it can cause several diseases in those regions usually, but they won't usually cause diseases. They will only cause disease if there is a compromised situation all the time, if there is a compromised situation going on. And the compromised situation, it can be of a normal immune system which is weaker immune system. So just start of the immune system, or what we can say unprepared immune system, So this is a kind of compromised situation, unprepared immune system, which we can find in, let's say, which we can find in infants, right? We can find in infants or newborns. Or on the other hand, we can have immunocompromised situation. That means immune system is not working properly due to the taking of immunosuppressive drugs, right? So immunocompromised system, and in those case, so immunosuppressive, so let me write immune suppressive drugs will be there. So if the effect of immunosuppressive drug is there, then we can produce immunocompromised people where this kind of infection will be common. Otherwise, in healthy organism, a healthy individual, we won't see this kind of infections taking their toll most of the cases. But all the infection we have talked about, skin infection, eye infection, we have talked about lungs, infection in brain, and also there is infection in, I forgot to mention, infection in urinary tract. So infection in, so let me write here, infection in urinary tract. Now in this case of urinary tract infection, this is a result of again nosocomial infection or hospital acquired infection. Now in this hospital acquired infection, uh, so hospital acquired, right? So let me write nosocomial here, nosocomial. Now, why should anybody have a kind of nosocomial infection related to urinary tract, right? Because remember, there people when they are catheterized in some extent during the surgical approaches and other approaches, in those conditions, if this catheterization is not accurately maintained, and if there is any way of the contamination of Pseudomonas aeruginosa there, it can cause urinary tract infections. And all the time the infection that we're going to see, here you can see in the skin rashes and pustules, eye ophthalmitis, conjunctivitis, in all these cases, it ultimately relates with and take everything to two important cores. One is inflammation. Let me change the color here. So one is inflammation. So let's take this one, say this one. So one is inflammation, and another one is the necrosis. Change it. Another one is necrosis of tissue. So ultimately they lead to two different conditions. One is inflammation and then necrosis of the tissue, right? Inflammation is required, that means they are activating the immune system cells, because our own body system will try to protect ourselves, so that's why this inflammation will set. And necrosis is caused by this bacteria, because necrosis will damage the tissue and help them colonizing much better way. Okay, so all of this infection that we have talked about so far, all of them here, all of them will be the part of, so let me write, these things are the part of acute kind of infection, acute phases of infection. But this kind of infection can be of systemic type. So let me write, this can be of systemic type. But the systemic infections reflecting the spread of organisms include bacteremia. Okay, but these are very uncommon, very uncommon, because this kind of systemic infections by Pseudomonas aeruginosa can only be seen in immunocompromised people, where the immune system is very poor. Otherwise we won't see these things, much, right? Now in this case, what we are talking about, that in this case the secondary pneumonia can be there, and then this spread of this microorganism from one place to another place, and finally when they reach the bone and joint. So what we get here, bone and joint, so bone and joint infection, sorry, bone and joint infection can be there. And also what we can get here is, here urinary tract infection can be there, or secondary, so let me write, secondary pneumonia, secondary pneumonia can be there. and obviously urinary tract infection, several types of urinary tract infection can be there, and one of those infection is endocarditis. So let me write endocarditis, let me write endocarditis. So this is a type of infection that could be there. And and the infection can also move from the different tissue level into the bloodstream, and as well as CNS or cerebrospinal fluid. So that is dangerous, because cerebrospinal fluid is pretend not to having any bacteria, because this is free of bacteria. This is a kind of sterile fluid in our body, but in these cases it can occur kind of bacteria from other sources in systemic infection. But these infections are very rare, can only be seen in immunocompromised people. Okay, so that's almost all the different kinds of infection caused by Pseudomonas aeruginosa, and I hope that's helpful. Thank you. So far, we have talked about the infectivity of Pseudomonas aeruginosa. Now, in this video, we'll be talking about the pathogenesis of them, right? So, let us talk about pathogenesis. So pathogenesis means, we know that pathogenesis means the way bacteria colonizes and infect an individual, right? Now in this case of this colonization and creating a infection, successful infection, Pseudomonas aeruginosa is having particular set of tools, and the tools that they are having, one is that the tool help them in attachment, and this tool here in this case, the tool is a pili. Right, so pili, which is a extension of a bacterial cytoplasm, which will help them to adhere to the host cell surface and then finally diffuse through the cell. Not actually diffuse through, but actually engulfed by the host cell via the endocytosis pathway. Now the second important thing that helps them to colonize there, the second important thing, right? Now this helps them to create a successful infection is secretion of a certain kind of toxins like exotoxins, and also it can create some endotoxins too, because we know that this Pseudomonas aeruginosa is a kind of gram negative. So let me write, it's a kind of gram negative microorganism. Now as they are gram negative microorganism, they are having a lipopolysaccharide layer outside. So if I draw the structure pretty carefully, what we can see is that, let's say this is the microorganism, and surrounding this we are having a kind of LPS layer. So let's do it simply like so. It's an LPS layer. Surrounding this LPS layer there is a kind of capsule. Now this capsule is made up with glycocalyx, or it is also called as glycocalyx layer or capsule. So let me write capsule. Now this glycocalyx layer or capsule is very important region. It is helping them to evade the host immunity, right? So for a successful pathogenesis of every individual bacteria or pathogen, there is two important tasks to accomplish. First important task is to colonize the host tissue or host cell, and second thing is that to escape the host immune system, right? Now the first thing is achieved using this pili, and also using exotoxins and endotoxins secreted by this particular type of bacteria. And the second one is to elude the immune system or escape the host immune defense. And the second important task of escaping the host immune defense is brought here using this layer. So here for the escape, so let me write escape of host defense or host immunity, there is the presence of this capsule. Okay, and capsule is also a kind of virulent factor for this bacteria. Okay, now in all these cases, so we are having here the toxin layer, here we are having and also there are pili coming out from different regions. So pili means this cytoplasmic extension that are coming out. So these are pili eventually. So using all these three important things, this bacteria is a master tool for colonizing the host cell. Now except for that, there are certain factors that are elaborating and that are enhancing their ability to colonize into a host cell and infect a particular tissue, is that the Pseudomonas aeruginosa, these things are very much kind of heat tolerant. So kind of heat tolerant. So this heat tolerance nature will allow them to be stayed for more longer period of time in host body, right? So that's a very important part, because in human body there is a core temperature which is a kind of high. In those conditions, this bacteria like Pseudomonas aeruginosa love to be present at this particular type of temperatures, right? That's an important factor. And also another important factor is that they are having a flagella, polar flagella. So let me draw a polar flagella. That means they are motile in nature. So this motility or motility is also helping them to colonize and move from one place to another place pretty fast. So let me write it here. So that means they are having a faster movement, right? Fast movement there. So these two are very important things. These are accessory things which are helping to further colonize this bacteria in host tissues, right? And also this they can easily evade through our immune system, and also they are secreting some exotoxin and endotoxin. Now the endotoxin part, which is commonly known as LPS or lipopolysaccharide layer. Now this LPS layer, that is denoted here with this green colored layer. Now this LPS is a kind of cell membrane protein. Especially this is the outer membrane or third kind of cell or external cell membrane protein. Now this cell membrane protein can act as a toxin when and only when the bacterial cell is degraded. So in certain conditions, some of the bacterial cell will be degraded themselves. They are suiciding, killing themselves, and finally this particular segment of LPS is released out, and they can act as an endotoxin to damage the host tissue. Except for that, they can cause several different types of toxins that will eventually damage the host tissues in different regions, so that this bacteria can easily make entry through those different regions. Okay. So these bacteria are equipped in all the ways, right? And they can stay for a longer period of time, and obviously they are very uh, faster in the movement. So all these things providing the potential pathogenesis move to this bacteria, so that they can cause several round of diseases. Now usually they are not, I've already told you that they're not a kind of typical pathogenic bacteria, but they can cause pathogenicity if they are getting the right time and right environment for them, right? So that's about the pathogenesis, and in the future video we'll be talking about a little bit about the treatment of the Pseudomonas aeruginosa. Okay, so that's it, and I hope that's helpful. Thank you. Okay guys, so we have already talked about the infectivity and pathogenesis of Pseudomonas aeruginosa. Now in this video we'll be talking about a very faint idea about the treatment of Pseudomonas aeruginosa infections. Now usually Pseudomonas aeruginosa is a kind of opportunistic pathogen, we all know that. So opportunistic pathogen. Now as they are opportunistic pathogen, usually all the kind of infections that we have already talked about regarding the Pseudomonas aeruginosa are originated from nosocomial. So they are of nosocomial type infection. That means the infection, all these different infections are hospital borne, right? So as the infections are hospital borne or hospital acquired in kind of infection, now usually the infections are associated with let's say the different surgical instruments, contaminated surgical instruments, right? Surgical instruments, or let me write contaminated with this red, surgical instruments. So contaminated surgical instruments can carry the Pseudomonas aeruginosa as a pathogen, which can cause infections there, right? So the processivity of the surgical instruments after each operation in operation theater is very important. So usually rest or most of this infection can be prevented. So let me write, this kind of infection can be prevented. So can be prevented if we what you can say, process this. If we process, so let me write, if we process the surgical instruments in the interval of different operations there, right? Because this kind of Pseudomonas aeruginosa can live at a particular temperature for a longer period of time. So if we sterile our equipments for a longer period of time at a particular temperature, above temperature, high temperature, we can get rid of this bacteria, and in those cases we can low down the nosocomial infections, right? Otherwise, if there is infection in onset, in those cases we are always having antibiotics to treat it, right? So we are always having antibiotics. Now, among the antibiotics place, what we are having there, we are having, let's say, we are having beta-lactams or penicillins. Right? So, in penicillin places, you can use penicillin to treat it, right? Beta-lactams to treat it. Also, we can have cephalosporins, third generation cephalosporin, like uh, ceftazidime. So, this is a kind of new cephalosporin name we have heard now. Cefta. So, let me write ceftazidime. So this is, or ceftazidime, whatever. So the ceftazidime is a cephalosporin type. So let me write it. Yes, it's a kind of cephalosporin. Okay. Usually we can prevent most of the infections by simply processing the surgical equipments as well as processing our food and all these things perfectly. If the infection is onset, we can use this penicillins or this cephalosporin tablets, and also we can use tobramycin. So let me write, we can use tobramycin. And if you're wondering from which part this tobramycin is coming, they are a type of aminoglycosides. So let me write this again to aminoglycosides. Okay. And also we can use ciprofloxacin from fluoroquinolone. So ciprofloxacin is also there. Right? So we can use all these different types of antibiotics to treat if the infection is onset. Otherwise, prevention is always better than cure. So most of the cases, the prevention measures are to be taken by the hospital authorities to clean and get rid of this kind of Pseudomonas aeruginosa, and not only Pseudomonas aeruginosa, because there are a lot of different bacteria that are coming during that are the causative agent of nosocomial infections. For prevention of those infections, you must sterile your equipments during the interval of different operations, and that's a very very important truth you should maintain as a good hospital habits. Okay, and also for the surgeons, and need to take care of the patient, and washing hands and everything, it's very very necessary during these times. Now in better conditioned hospitals in better countries, there are very very less number of cases regarding Pseudomonas aeruginosa case, but in developing countries where the hospital system is not that much strong, in those cases you can have this kind of infections. So again, you need to be very careful in the hospital areas if you are talking about Pseudomonas. In other cases, if it is onset, you don't need to worry about very much, because we are having antibiotics to fight against it, and it is a kind of sensitive to all kind of antibiotics that we have talked about. So nothing to worry about, and that's how we can treat it. And I hope that's helpful. Thank you.